All right. Thanks for coming. Thank you, Peter, for organizing this. I'm going to be, my name is Gaurav Patel. I am not Jessica Lee. I'm going to be presenting Jessica's results from our lab as she was unable to make the conference. And I also landed about 4 hours ago from New York, so if I become incoherent, feel free to holler and snap me out of it. Okay, so we are going to be talking about some work we've been doing in our lab looking at how multimodal signals may be integrated in the temporoparietal junction and the superior temporal sulcus, and how that's disturbed or disrupted in schizophrenia. So we have no disclosures to report. However, we do request that no one take any pictures of the slides. So a major focus of the lab is to look at social functioning in schizophrenia and to try and understand the neural mechanisms of it. So unlike the psychotic symptoms, which most people associate with schizophrenia, and those can be generally treated with medication, the negative and the cognitive symptoms, no effective treatments exist for those, or at least no clinically deployable versions of those treatments, and they contribute a significant amount to the disability in schizophrenia. So we're talking about people who are on medication but are still unable to hold jobs, have relationships, et cetera. So previous study from our lab, we used movie watching to look at naturalistically evoked brain activity in comparison between healthy controls and schizophrenia participants. These are, the schizophrenia participants here are chronically ill individuals with schizophrenia, mostly on medication, mostly outpatients, or all entirely outpatients. What we found was that when we compared schizophrenia and healthy controls, oh, that red dot's weird, okay. What we found is that that in the temporoparietal junction right here, that there was decreased typicality of the movie evoked activity compared to healthy controls, and as measured by intersubject correlation. And the intersubject correlation of that region correlated with a social cognition test called the TACIT, the Awareness of Social Inference Test, which is a movie-based test of how well do people understand social situations depicted on the screen. It was actually developed in Australia about 25 years ago. So in a follow-up study, then we looked at the functional connectivity of all of these areas. So what we've done here, each of these ROIs here is, rather than using one of the resting state parcellation schemes that are out there, we used a bunch of tasks to functionally localize brain areas so that we could understand which brain area is doing what function in relation to social cognition. So when we look at the functional connectivity of all those ROIs, we get, we see that in general, that the resting state functional connectivity between healthy controls and schizophrenia is roughly the same. You see in both populations this kind of large chunk here with the orange being visual areas, attention, prefrontal, singular percular areas here, and then these yellowish areas are theory of mind / default mode areas. And you can see that those are kind of opposed. And what we see then is that there's another set of areas that connect the visual areas to the theory of mind areas through the posterior superior temporal sulcus and the temporoparietal junction here. And we see that the connectivity of those regions specifically is decreased in schizophrenia. And the degree to which it is decreased, the strength of that connectivity in controls and in the healthy control population correlates with how well activated the temporoparietal junction is during the movie, but not in patients, suggesting that the integrity of the connectivity of these regions is important for processing those naturalistic stimuli. Okay, so that was the background. Now, what we did here was, like I said, we looked at how resting state functional connectivity, which kind of roughly approximates, let's say, anatomical connectivity and how much brain areas are used together. we looked at how well that predicts movie watching activity, the typicality of it as measured by intersubject correlation. What we didn't do was directly look at how well the functional connectivity at rest relates to the communication between areas while people are watching these movies. So, and that was of direct interest to us to better understand what this relationship may be. So we went back and reanalyzed 3 data sets that we had in the lab to look at this directly. And in addition, because the movies do involve auditory stimuli and obviously language, we added in ROIs from Matt Glasser's parcellation scheme to, since we didn't have localizers for those, to map out those areas as well. And this is the resting state functional connectivity on the left of all of those ROIs, and this is just a schematic of how they might be connected. And what you can see is that this resting state graph roughly approximates the areas as, or the gradients of areas as described by Daniel Margulies and others, with the early visual areas on one side, auditory areas on the other side, and the default mode / higher order cognition areas on the opposite side of the graph. What you can see here is what we had seen previously about how visual information, may get to these higher order theory of mind areas to support these social inference operations. But now with the auditory and language areas, you can see that there is a a path from auditory cortex through the language and STS areas. Critically, both of these areas overlap in the STS, leading us to think that maybe this is a site of some type of auditory and visual integration in the support of social inference. So as I said, we went back and analyzed these data sets, which were collected on a GE3T scanner, roughly 20 to 25 subjects who either watched a silent movie, 15 minutes of the good, the bad, the ugly with no audio track. They listened to an auditory story, or just listened to a radio story called Pie Man, which has often been used by Uri Hassan and other people for these kinds of experiments. And then they also watched, about 12 minutes of BBC Sherlock in the scanner. All of these individuals also had resting state functional connectivity, usually about 22 minutes of resting state functional connectivity. And what we're going to do here then, as I'm going to show you in a second, is compare the movie evoked functional connectivity versus the resting state functional connectivity. So just to give you some details, we pre-process these data through the human connectome processing pipelines with some additional post-processing for movement artifacts, including frame censoring. And functional connectivity is very simply just the correlation of the brain activity at rest, or in one of the movie conditions, the correlation between 2 ROIs. Based on the based on the graphs, we then calculated the weighted shortest path length between ROIs at multiple thresholds from the 60th to the 90th percentile in 5% increments. And then what we're going to be showing you is the average of all of these SPL calculations across all of these percentiles. All the results are P.05 FDR corrected by permutation testing unless otherwise indicated. And just for ease of comparison, we are going to be showing you the schizophrenia data overlaid on the healthy control data just to make things easier to compare. Okay, so we are going to be looking at, how are these connectivity patterns shown here at rest, how are they going to change as a function of the movie input? So as we run through this is now what the network graph looks like when people are listening to a story. And you can see that the distance in the graph between the auditory areas and these high-level areas decreases a bit more than the visual. This is now when they're watching the silent movie. You can see kind of somewhat of the opposite effect. And then this is when they're watching the combined movie. You can see some averaging of those two effects. So the key thing here is that there are large differences in the functional connectivity patterns under these conditions. So we're going to quantify that now, as I said, by looking at the weighted shortest path length to either A1, right A1, primary auditory cortex, or right V1. So for the auditory-only stimulus, we're going to calculate it in relation to right A1 for every ROI, and that's what this map looks like. And the ones that are colored here are the ones that have a significant change in path length while listening to the auditory story versus rest to A1. This is what it looks like then for the visual areas, right, in comparison to V1 for the visual-only movie. So combined, what you can see is that while there are modality-specific, changes in the functional connectivity during these conditions versus rest, that there's a set of areas up here in the upper right corresponding to the STS and the STG areas and the temporal junction that overlap between the two. So to highlight that, we added these two weighted shortest path length changes. We added them together to create what we call the AV sum metric. And what you can see here is you're highlighting the areas in the STS and the temporal junction. These are the ones whose path length changes most depending on the auditory or the visual inputs. And this is what it looks like on the brain. And again, you can see it's in the temporal junction and STS is where most of the significant changes are. So now we can do this also for the multimodal movie, and you can see a very similar pattern between the two, that there is changes in the STS. Those are the source of the biggest changes in shortest path length. So looking now at the schizophrenia participants, if we look at that AV sum metric, this is what it looks like in schizophrenia versus what I already showed you in the healthy controls, and this is the difference. And what you can see, the biggest differences are in the visual areas and the auditory areas, and then up into the posterior STS and the STS, along with some dorsal attention network. Again, this is what it looks like on the brain. Again, there's the STS areas, some visual areas, some auditory areas, and a couple dorsal attention network areas. When we break this down then by which ROI shifted the most towards auditory, primary auditory cortex in green versus primary visual cortex in blue, we see kind of an interesting pattern here, which is that visual areas over here on the bottom left shift towards, in schizophrenia participants, shift more towards auditory cortex than healthy controls. And conversely, auditory areas shift towards visual cortex, right? So this is essentially saying that when someone is watching the silent movie, there is increased synchronization, if you have schizophrenia, of your auditory cortex with V1 than healthy controls compared to rest. And you can just see this highlighted here when you look at the modality-specific differences, that auditory cortex gets more visual changes, auditory stimulus gets more visual changes, visual stimulus gets more auditory cortex changes. So in conclusion, what we've shown is that the functional connectivity structure at rest versus under naturalistic stimulation differs strongly, with the largest reorganization in STS of both auditory and visual stimulation. The increased synchronization between non-simulated and simulated sensory areas in schizophrenia participants may indicate some type of maladaptive failure of the STS to segregate or appropriately integrate sensory processing streams in schizophrenia. And in general, that if examining the differences in functional connectivity patterns at rest versus these various stimulation conditions can reveal information that examining one by themselves, any one of those by themselves, would not reveal. So the future direction is to, of course, replicate these results in a larger sample size, which we're working on right now, but also on the other side is to use the precision functional mapping methods advanced by Evan Gordon and others to look at this more directly within the individual and see, do we see these changes at the ROI specific level? With that, I'd like to thank Jessica, who of course did all the work here, also Tammy and Ahmad, who are here, who helped with the conceptualization of this early on, and the rest of the lab here, and happy to take any questions later. Thanks.